Good morning, good afternoon to our audience, and thank you very much for joining our webinar on the analysis of veterinary drug residues and food of animal origin. So my name is Giulia Rosar. I'm the product manager of Eurofins Technologies, and today I will be together with a portion of my team because unfortunately I have one that is missing. We will have a short introduction on uses and misuses of veterinary drugs, an overview over the available technologies for residues analysis in food of animal origin, and then a section dedicated to the evaluation and validation of test kits, and finally, good practice for internal and external quality monitoring during routine analytical implementation. So let me thanks anyway my teammates, both of them. But I believe that just Francesca is available. Unfortunately, Evelina is sick. Hello, Francesca.
Thank you, Julia, for the introduction, and thank you to the audience for joining.
So I take a final moment for some technical aspects for the attendees. I would like to remind that you're all muted, but the chat box is always open. So please share your comments and questions, and we will read as many as feasible at the end of the talk. So let us start. A very simple question to start. What are veterinary drugs? Veterinary drugs are any active substance administered to animals for the treatment of diseases, as logical, but also for other purposes like the prevention of diseases, the improvement of growth and productivity, and the modification of physiological functions. There are several categories of compounds that can be administered to animals, including, I don't know, anti-tumor drugs, tranquilizer, anti-inflammatory, anesthetics, and most relevant, the antimicrobial drugs, including antibacterial, coccidiostats, and antihelminths. Depending on the scope, the dosage must be calibrated. So for growth promotions and prophylaxis, these are achieved with low concentrations of medicines. The quantity must be increased for one efficient metaphylaxis when first sickness symptoms are detected. And then the highest dosages are given to treat real sick animals. Residues of drugs given to animals may be present in a lot of foods, including meat and poultry, milk and dairy products, eggs, fish, and other aquaculture products, and also honey. Substances may accumulate as native compounds or in metabolized forms. They can be free or bound to tissues and may remain in the food for a certain time after the completion of the treatment. Drug residues in food may lead to acute bad effects in the consumers, such as allergic reaction, or, more concerning, I have to say, long-term health problems, such as antimicrobial drug resistance, but also cancerogenity, mutagenicity, bone marrow depression, and disruption of our normal microbiota. It is therefore clear why multiple jurisdictions around the world have responded over the past few years by restricting certain drugs in livestock, bees, and aquaculture. Some compounds have been forbidden for the treatment of food-producing animals because, I don't know, for their severe adverse effects, for instance. But also there are forbidden practices somewhere. Growth promotion has been banned in the European Union since 2006. And a large number of Asian countries have also already banned or are in the process of banning the use of antimicrobial growth promoters. Also, several countries have banned the treatment of bees with antibiotics, and honey must be antibiotics free. Since the past few years, both the World Health Organization and the European Medicine Agency have pushed on the reduction of drugs in use in animals. So I'm asking Francesca, how is the current situation with the use of antibiotics and active drugs in animals, and what about controls?
I have to say, levels of use vary a lot from country to country. For example, some Northern European countries use very low quantities of drug to treat animals, as reported in European surveillance reports. Also, the FDA a couple of years ago showed a reduction of use of antimicrobials in livestock. I have reported here two screenshots from European Food Safety Authority and the Food and Drug Administration. The situation is known to be different in other regions in the world, although official data about the quantity and pattern of antibiotics used in husbandry and aquaculture are not available. According to the FAO, little more than 40 countries in the world collect agricultural antimicrobial use data. As you were mentioning, Julia, the concern over antimicrobial resistance related to the use and misuse of drugs in husbandry and agriculture is a priority for the World Health Organization and led the European Medicines Agency to provide with a categorization of antibiotics for animals in their care, dividing substances in four classes: to be avoided, restricted, to be used with caution, and to be used with prudence. I won't go through this slide, but it's interesting to us today to see what the categories include, which are the categorization basis, and start to get familiar with the names of the antibiotics families. Given all the recommendation and responsible use over antimicrobials and limiting certain practices, both the Food and Drug Administration and the European Union, as well as several Asiatic countries, have listed the regulatory limits for a number of residues, defining hence the maximum tolerance concentration of each compound in each animal portion, with differences among species, and a special attention to animal products like eggs and milk. In this slide, we can see just the very beginning of the list, with a few compounds listed only, but you can ask for the comprehensive document to Eurofin Technologies.
Francesca, are there limits or indications or somehow, let's say, tolerated concentration for forbidden compounds?
Well, forbidden compounds should not be present at all, so we cannot refer here to permitted concentration or maximum limits. In some cases, regulation have issued some action levels, point of action, or minimum performance sensitivity concentration to be ensured at least. In this slide, there are the new reference point of action for non-allowed substances in European Union, as reported in the recent Commission regulation 2090-8071. It is not important to mention that these levels are not to be intended as safe quantities that are permitted. As in the regulation, it is clearly reported that the detection of residues of prohibited substances, even below established reference point for action, could be a signal of misuse of such substances. Who is the committed to run the analysis for antimicrobial and drug residues in food of animal origin along the production chain. Verification could be run at the farm site to monitor sometimes the efficiency of the withdrawal period or to search for accidental contamination with drug. Official control can also take sample directly from the farm, both for surveillance purpose or for research project. Along the transformation and distribution chain, industries and retail could ask arrange and be controlled both for the compliance of meat and product to the regulation, as well as to some specific antibiotic-free claim. Sometimes industries could have specific needs also from a process point of view, as some residues could interfere with transformation, as cheese fermentation. Controls are often arranged by imported good also.
So thank you, Francesca. We have learned very generically who is performing the analysis and where. Now we have to understand how. So which techniques are available? Multiple technologies are commercially available and described also in the state of the art for the analysis of meat, kidney, liver, animal urine, eggs and milk, honey, fish, and other foods of animal origins for the presence of drug residues. Inhibition assays are really inexpensive tests for the multiple detection of some families of antibiotics, typically in milk and some meat samples. The tests are made of a medium. I don't know if you're familiar with it. This medium contains bacterial spores and a color indicator. So when adding a sample that contains antibiotics, the color change is prevented and turns to be a positive, non-quantitative alert of contamination. Ease of use and cost efficiency are coupled for this kind of assays to a partial coverage of antimicrobial detectable by such kind of assays. I mean, we cannot know what is present in the sample, and we cannot cover all the drugs that could be used for animals producing milk or for animals whose meat is eaten. Immunoassays are available actually in several formats, like fast lateral flow devices that are typically used for the milk analysis, and plate-based or other planar arrangements that rely both on one enzymatic chromogenic reaction or a chemiluminescent one. As veterinary drugs are really small molecules, all these immunoassays are competitive, with signals that are inversely proportional to the concentration of drugs in the sample or in the standard use in the calibration. Through sensitivity enhancement and antibody engineering that led over the past few years both to very specific as well as very broad range immunoassays, binding assays are the option of choice for the food industries. And in plate format, I believe they represent still a reliable ally in the screening of high analytical volumes in public and private laboratories. Immuno. Binding reagents used in the immunoassays, despite the specific formats, let's say, are antibodies. But the same layout and designs, as I mean strips, plate-based assay, and so on, can be applied to other binding molecules, such as receptors, aptamers, or molecular imprinted polymers. So, last line of the slide. Liquid chromatography coupled to mass spectrometry, among other instrumental techniques, has become a relevant technology because of the high sensitivity and specificity of detector that allows to apply, very generic sample preparation procedures, which simultaneously detect, extract, a wide variety of residues with also different physical and chemical properties. It is clear to understand that these 5 options and multiple options have different features in terms of performances and in terms of cost. So let me stay a little bit on a very important point. We have already mentioned that hundreds of compounds have been regulated and have been restricted somehow in their use on animals. So how to cover efficiently the huge number of residues that could be eventually present in one piece of meat, one egg, or one milk sample. This is not the case, like allergens, where we have to search in the maximum and the worst scenario for 14 substances, or mycotoxins, where we have just 7 regulated compounds. We have hundreds here. So what are multi-residual screening test kits? Do they exist at the state of the art, and which are the approaches? There can be kits that are able indeed to detect several compounds in one run, having one sample preparation that extracts consistently all of them. And different binding reagents or different detectors are used to detect the different molecules without mutual interference. So the presence of one should not interfere with the presence of the other one. Sorry. In this slide, we reported the representation of a multiple lateral flow device on the left with 3 independent test lines for 3 different compounds. In the center of the slide, there is one planar array or planar multiplex, having a spotted surface with different binding reagents for capturing different compounds. And finally, on the right side of the slide, we have represented one suspension array, where there are color-coded beads that are coupled with specific binding reagents like antibodies, and these beads are mixed together with the sample to detect multiple residues. And I can tell you that yes, they do all exist at the state of the art, and they are also commercially available. Despite the technology and the detection system that we have not, let's say, discussed and managed in detail in this seminar, there are still a few degrees of complexity that must be touched, in my opinion. Antibodies, but also receptors or other binding molecules, can bind and detect not only one, but multiple compounds with different degree of selectivity, or in other words, different cross-reactivity percentage. Broad range binding reagents can be therefore used and are actually used for covering entire compounds families where drugs have similar chemical structures. So let us understand a little bit. This is an example of something that is actually existing, a strip with multiple lines. Line 1 detects a certain compound, line 2 a certain compound, but line 3 is for a family of compounds that probably are close, chemically speaking, one to each other. And in the kit insert in the manual of this hypothetical strip, we see that the capacity of binding is expressed in terms of percentage. What do these percentage mean? Let us think to inhibition curves. As we are speaking about, competitive assays, there is one antibody that reacts differently with 4 compounds named from A to D. A is the parental drug used to raise the antibody and is assigned with 100% cross-reactivity. We see in this generic graph with signal or relative signal intensity on the y-axis versus a generic range of concentration, the A curve is the bold one, okay? Compounds B and D have less affinity for the antibody and indeed lead to less sensitive curves displayed on the right side of the graph. You know, those that are dark blue, let's say. That means that we need more compound B and D to have the same level of inhibition, the same level, the same signal level than compound A. But on the contrary, the pale blue curve that is on the left is obtained by the compound C. It shows somehow better affinity for the antibody and high inhibition capacity. And this is a realistic situation, probably because compound C is closer to the substance and the immunogen used to raise the antibody, for instance. So what does this mean? What should we really expect in terms of performances, in terms of response from one assay having these cross-reactivity patterns? And this is just an example. Broad range antibodies and receptors have been widely used in the past, I don't know, 10 years or something, to develop assays that are capable to detect a whole family or a portion of a family of drug residues. And it makes sense indeed, not to search for every single, I don't know, sulfonamide, every single tetracycline in one sample, especially if we put in the shoes of official control bodies that are running surveillance and monitoring programs. Why should they use, I don't know, 10 kits, one for sulfametazine, one for sulfathiazole, one for another sulfonamide, for each meat sample, each milk sample, and so on. So accessing broad range assay is a key to keep the analysis cost efficient, but still reliable. So we have to point out that in this case, we have to think that the result cannot be fully quantitative, as cross reagents and mixtures cannot be discriminated, and the result could give just semi-quantitative indication. We cannot think that compound D having 33% of cross-reactivity will lead exactly to 33% recovery. That is not true. Also, a lot of time, as in this scenario, we access to limited information, like compound C has more than 100% cross-reactivity. But what is more than 100? It is 110% or it is 600%. We cannot correlate exactly the result, the number that is given by the assay that typically refers normally to the major compound, that in this case in compound A, and calculate exactly the recovery based on the cross-reactivity. This is a not recommended approach. We have to accept that multi-residual broad range assays lead to simply semi-quantitative results. So, veterinary drug screenings measure what counts? What should we take into account when selecting a device for the analysis of veterinary drug residues in food? We have to evaluate what counts for us. Every company, every laboratory should really list their requirements in terms of targets. What do we want to determine? Are we interested in getting a warning? For instance, if any medical treatment was done, or do we need to know if some permitted compounds are above maximum residual limits? Or are we searching specifically for some forbidden molecules that must be equal to zero? How important is the capacity of the assay to distinguish very small concentration of the contaminant? On the contrary, how little important is the recovery and the accuracy for the quantification of a forbidden compound? We have to think to these questions. In my opinion, it doesn't make any sense to search for a very sensitive screening for, I don't know, sulfonamides that are permitted with several ppbs that are permitted in milk, in meat, and so on. On the contrary, we have to search for, I don't know, ELISA test kits or methods for chloramphenicol with boosted sensitivity because chloramphenicol is prohibited. And other than performances, in light blue, there are other 4 topics. Do we care about these aspects? Time to result, is that important? Sample preparation complexity, is this important? What about the supplier reliability, time for the shipment, technical support provided, and so on? These obviously also could drive the evaluation, let's say, over the product also. Once we have selected a product and we have to validate, we have to define for the very beginning, what does validation mean? To validate means to verify that the assay performances are compliant with the specifications given, and does not mean that the assay is good. Okay? A validation report should always be obtained, in our experience, by the kit producer, by the kit developer. And this is supposed to contain both data collected internally and also through, if available, through some collaborative study or independent evaluation. An end user that is well equipped and trained can definitely also run a validation protocol, having access to materials and instruments for reference analysis, not to risk to run a bare comparison of one screening to another. This is not typically a validation. This is something different, let's say. How? A few guidelines and outlines are available for the validation of screening test kits for veterinary drug residues. The European Union, for instance, has recently released a new regulation, this was for the last year, that applies to official controls and describes exactly the performances and the acceptance criteria for any analysis performed on veterinary drug residues and food of animal origin. So, European Commission 2021-808, let us enter exactly what does it prescribe. This is the table that lists the performances that must be validated when a method has an official purpose, but it is really good for any purpose, depending on its scope, whether it is qualitative, semi-quantitative, or quantitative. For all the screening methods, You see, it is recommended the verification of selectivity and specificity. This means that we have to know how and what can interfere in the assay. That is really important. What could affect the measurement of my target, my analyte that is included in the matrix, or if there are any congeners, any related compounds that could be detected by the assay. Also, clearly, the recommendation is to validate the stability and assay robustness to small, mistakes, deviations. And there is a recommendation to validate, you see, for the screening methods, always the CC beta. While accuracy and precision, that sometimes we really like this concept, for the very first time, they are indicated to be validated, but only for the quantitative methods. In the previous version, the accuracy was not demanded, was not required for screening methods, just for confirmation instrumental analysis. CC beta. What is this parameter? What does it relate to? Does it have any synonyms we are more familiar with? So, the definition provided by the European regulation reports the CC beta to be the detection capacity of the screening, meaning the smallest content of analyte that can be detected or quantified in a sample with a narrow probability of beta. And what is this? The beta error should be equal to 5% for both authorized and prohibited substances. Making the long story short, this parameter represents the matrix-based sensitivity. It is the lowest concentration. We're able to demonstrate that the assay is capable to detect 95% of the times, meaning that if we have 20 samples, very low contaminated, we can accept that one of them is detected as false negative, false compliant, and 19 are captured, are identified as positive. This is the concept of CC beta. We are not going to read all these sentences, but for authorized and for forbidden substances, the European Union lists the multiple options to calculate exactly this CC beta. But as complex as this may seem, we have to figure it out how to really calculate and determine this value. The calculation of CC beta in any case relies on the analysis of spiked samples. This is something very important. I am aware that there are a lot of approaches for validation that calculate the sensitivity of one assay on the basis of the mean response of some negative samples or some zero standard only. And through the standard deviation, there is an hypothetical assessment of the limit of quantification, LOQ, or other terms. In the European regulation, also in the past versions and guidelines as well, it is prescribed to use multiple independent fortified materials. Practically speaking, this means that we need multiple independent samples, at least 20. We need to know for sure that are blank, either because they are from animals that we know for sure that have not been treated and not exposed to the substance we are running the analysis. Or, even better, we know that they are blank because we can access a reference analytical method with suitable sensitivity to prove that they are not contaminated. So we need 20 samples, we need the assurance that they are blank, and we need something more. We need a spiking solution made of an adequate standard. This is what we need. Okay? So, a very generic environment. This is not related to a molecule specifically, but just to have visual depiction. Blank samples should distribute around one value. That could be expressed in terms, I don't know, of apparent dosage, or better, like in this example, of relative signal. That in the ELISA is typically B over B0, and lateral flow could have different values, T over C intensity line, or some other way of calculating their signal. Anyway, those samples that are spiked at the CC beta concentration should represent a very distinguished population of data with adequate, consistent delta difference to the signal of blanks. So in this example, we see that, maybe I can drive you through the arrow. One determination is indeed overlaid to the population of blanks. That is representing the single false compliant result permitted out to 20 independent determination. Okay, so we see here the blank samples with low inhibition in terms of relative signal, that have a certain scatter distribution. This is normal, and we can see just using multiple independent samples. And we see the CC beta spiked population. One is not detected. This is acceptable, and the rest are consistently detected. Detected or quantified, it means that it is not intrinsic in the sense of the CC beta, the quantification concept. The important thing is to distinguish this concentration from blanks. Okay? Studying the behavior of blank materials is absolutely important. I would say that in our experience of 20 years and more, it is fully mandatory. It's a key part of the assay validation, and we typically run in parallel with CC beta experiments. Depending on the composition of the matrix and on sample preparation protocol and complexity, there could be some interference with the binding sites of the antibodies or receptors used in the assay. And this is something that must be characterized and handled. So let us start with a very simple example of one ELISA, where blank materials did not show any matrix effect. We see in the graph on the left, we have 20 determinations in dark blue, almost with no inhibition, that are blank matrix. And on the right, you see the mean value with the standard deviation that is very small. The pattern of inhibition obtained with spiked samples reflects the variability already recorded from blanks. That is a very good thing, with samples that are systematically below the blanks, with consistent difference, with consistent delta and compact results. What is this? An ideal scenario, obviously. So keep your eyes on the screen. What's changed now? The matrix effect recorded for blanks for this second immunoassay is much higher. Non-contaminated materials produce, for some reasons, I don't know, a higher inhibition. And for sure, some apparent results that must be handled. The decision limits or cutoff value are recorded, okay? This means that routine results of unknown samples that are less inhibited than this red line, that is the decision limit, should be considered safely as negative, as compliant. CC beta spiked samples are still well separated from length, as visible in the pale blue series of data. And there is just one, let me highlight it, probably false negative result, as this is acceptable. So you see how important it is to determine the behavior of blank materials accessing for real to a huge number of independent samples, not to replicate 20 times the same blank material or the same blank extract, please. Let us focus a little bit on metrics. What do we intend for metrics? Am I allowed to, I don't know, validate fish and shrimps together because they are all seafood? Bovine and swine urine together because they are all urine? How in details should I go? How many validations should I then run? This is an important thing to know also in terms of costs and times and investment. That will be the assay itself to guide you finally. Let us have a look at this case. We have 20 determinations, but this is not one matrix, I can tell you. 20 determination led in this scenario to two populations and a non-normal, non-Gaussian distribution. And we see it both for blank and spiked materials. In this very case, we are watching this is a real case I took from our data, bovine urines of adult on the left and young animals on the right. This assay was intended for the detection of one hormone, okay? And we saw that sex and age of the animal, especially the age, led to different data population. It means that urines cannot be considered as one matrix. So remember that to treat the samples as one group and as representative of one single matrix, data cannot be bimodal. They should have a normal distribution and the reasonable dispersion. It happened also for some assays for honey analysis. It does not happen always, all the time, but we discovered for one of our assays that colored honey, those that are orange to red, could lead to different results than the clear acacia on flowers honey. We have spoken about matrix importance. Is the analyte important also? Yes, it is, especially if we are validating a multi-residual kit or a broad range kit. So left side, we have a planar array or a multi-line lateral flow. If we are validating a multi-residual assay, each of the targets should be spiked independently. And also, let me recommend to do mixtures to learn the assay behavior when drugs cocktail are present in the sample extract. This is to characterize and prevent any interference between binding sites. So it is really complex to validate multiplex methods. For broad range methods, there are basically two options. The validation, particularly the sensitivity assessment, could be run spiking the most relevant target or targets, those that appear more often in one routing sample, so that the performance of the assay is well predicted. On the other hand, it is possible to go for the worst reactant scenario, especially when testing the sensitivity, meaning the CC beta assessment. you select the molecule with the lowest affinity. You remember and this graph, you take the compound leading to this curve on the right, and you validate the assay sensitivity for this very molecule. It means that you can safely predict that same or even better sensitivity is ensured for all the other cross-reactant. It's a way to save time. So, to bring some very practical concepts in our hands. When evaluating a new screening product, it is highly recommended, please, to collect all the information from the producer. Let me reinforce the importance of asking and aligning with the kit supplier on the validation method applied and the concept included in the validation report and the instructions. Some informations could be misleading. Data could be presented differently by different companies, so it is important to get the right support to understand the document collected. There are a lot of other terms that are reported, especially in the manual of the kits, like limit of detection, limit of quantification, CC beta, someone validate also CC alpha. You have to normalize this concept. Okay. To run the validation, we do recommend to follow adherently the validation outline of the European Union, giving space to natural matrix variability and focusing on the sensitivity assessment rather than the accuracy and recovery experiments that for some extent have really limited value. It's not that we do not do these experiments, but it's really more important to know about the sensitivity of the assay. And besides spiking experiments, when available, try to use also some reference materials to assess, to confirm what we saw through spiking experiments. We skipped this part because we are running out of time, but it is really important. So it is not that valuable to verify the recovery of a forbidden compound. When you have in your sample, you already have a problem. It is not that important to check the accuracy of a broad range assay, because then in a real sample you simply don't know what is inside. And so really focus on searching these independent routine blank materials. And finally, we have to conclude. I know we have little time left this presentation with the section dedicated to the routine monitoring. Performances assessed once in a lifetime represent, yes, a solid background for reliability, but internal and external quality verification are good mandatory practice that must be implemented for an aware analytical management.
You're right, Julia. Indeed, every lab needs to have a strategy to obtain and maintain good quality standard. And to do that is crucial to understand how to verify the performance of an analytical session and how to carry out internal and external quality controls. Once a kit has been validated, its performance must be checked over the time, because an excellent kit will never give reliable result if it's not used in the correct way. Uncertainty is one of the aspects that a quality assurance program must consider and monitor. The uncertainty assessment must take into account the contribution provided by instrument, by the operators, and obviously by the kit. Let's see in details what we can do to assess whether our analytical session is complex or not. Assay specification are simple parameters that have to be checked before accepting any data related to the samples. Typically, they are referred to the maximum signal intensity and curve positioning. If a curve is present in the assay, obviously. If replicates are run, the average variability should be checked as well. Having a compliant test with the curve in the right position does not save the technician from accidental mistake occurred during the sample preparation. The key is to regularly use a reference material which must have a no concentration and an acceptable range of concentration. The reference sample must have the same composition as the routine ones and must be used exactly as the routine samples in terms of extraction and assay execution. I really want to make my point very clear and reinforce what should be avoided. Among other, the most important things are having a sample with a reasonable dosage with the dynamic range of the assay. Having a stable material with concentration given by a reference method. Use a homogeneous material, store it properly, extract all the time using the same quantity in terms of gram or milliliter you use for routine sample. Do not save the extract in fridge overnight to carry out the ELISA assay the next day, because it could be possible a transformation of the extract due, for example, to bacteria presence, which can affect final results. Here we report an example of control chart to track the collected result for a reference material used in routine over several days. Results are reported in a table, and for each session, the average of concentration of and the coefficient of variation are calculated to monitor the measured concentration and precision. Then values are plotted on a graph with concentration against the time. This graph is indeed called control chart. Ideally, the control chart should include two materials at different concentration. They can be used to monitor the quality of the execution over the time, the differences among operators, the kit batch change that should not introduce any difference in the assay performances, the day-to-day assay reliability. Prior ending one sample, a new one should be used in parallel. Is that really important to use multiple material for real? Yes. Well, graph on the top of the slide show a realistic situation. For one material, a trend leading to overestimation is recorded. If this was the unique sample implemented, then it would have been easy to believe that the assay was having some issues. But we see from the other two materials that the assay is there keeping a good performance. On the other hand, when one session suddenly causes aberrant results on both material, it is easy to understand that the session had something wrong. With one single sample in routine, dubs are more frequent, and this could lead the technician to repeat the analysis more than needed. In parallel with internal quality control, there is external quality assurance, which is performed conducting an interlaboratory test arranged by providers able to compare the results obtained from different labs. Samples are supplied as blind material, and each lab carries out the analysis following its own routine procedures. Results from all the participants are collected in a report, and an assigned value is given, either based on participants' consensus or on the basis of reference method. Providers calculate a score for each participant, which defines how the result is close to assigned value. When the zeta score is less than or equal to 2, the performance of the lab is considered acceptable. In case of veterinary drug residues analysis, some providers can substitute zeta score with judgments if semi-quantitative or qualitative results are given. Just thinking, do some multiple analyte contaminated material analyzed with a broad range screening. To build a proper and consolidate routine for proficiency testing participation is necessary to mark each deadline on the calendar and arrange to carry out the tests a little in advance. When the material arrives, store it properly, and when you decide to carry out the test, reconstitute the sample exactly as described on the provider prescriptions. On the contrary, the extraction procedure must be carried out according to the routine procedure. It is good to use another reference material you have already used as internal control and perform the analysis twice in order to evaluate both the recovery and the reproducibility of the result. Be careful to provide the result following exactly the provider's prescription. Otherwise, provider will reject the results. If you participate to a proficiency test for the analysis of forbidden veterinary drug residues, which may involve also the use of broad-range kits, it is good to provide the result as lower or higher than the CC beta value. In fact, with this kind of kit, it is not interesting to know the exact concentration, but it's important to screen positive and negative samples. In this case, the provider will evaluate the performance of the laboratory as satisfactory or unsatisfactory without providing a specific score value. The fundamental concept of internal and external controls is to regularly monitor the routine work of the lab, checking the work of the technician, especially new ones, the instrument, and the batch variation of the kit. This is possible by using good reference materials and participating to proficiency test at least once a year.
So thank you, Francesca. Very briefly, because we are running out of time, which is the take-home message we would like to leave you today? There are a lot of very good reasons to look for antibiotics and drug residues in food of animal origin. And a lot of technologies are available on the market, I have to say. It is good to take a little time to investigate pros and cons of each technology. But this evaluation must take into consideration the rate of importance of parameters like ease of use, time to result, cost, and knowing how many samples and parallel do we typically have to run. So you see here the rather graphs that represent more or less for each technology, which could be the benefits and also the limitation of each technology. There is not the ideal solution. It's just a map to have a guidance through different proposals. And every laboratory, every reality has to make the choice. Each of these technologies have indeed multiple kits available on the market. It is important to learn in advance their claims and performances. And then it is recommended to design a sustainable and still efficient validation plan focused on the metrics. Let us put in the center metrics variability, metrics effect, and so on. Searching for shortcuts that use a limited number of samples or some, mathematics to expect a certain performances could lean in routine, unexpected results, or some results that we are not so good in handling. And last but not least, we've learned from Francesca, the validation that we run at the initial time is not everything. Routine, little investment in quality verifications are the key to keep the analysis reliable over the time. So, knowing that we were not exhaustive and we would need probably 1 hour more to enter into the details of the validation, different approaches, the benefits of the guideline provided by the European Union, and so on, we have done for today. So thank you very much for joining us today. We are available to answer a few questions that I see in the chat box. So first question is, which judgments are given to VDR proficiency results? Judgments depend on the proficiency test providers. So we have experience with many companies that range. There are not so many, but we have some experience. I have seen, as Francesca was reporting, satisfactory when the result is in line with the assigned value of the sample. Unsatisfactory, that is given typically to false compliant result, because for screening test kits for veterinary drug residues, a test that is not able to detect contamination, well, this is not satisfactory. For false positive results, that yes, represent something that then you have to confirm through an instrumental analysis, but in the end, it's not a risk for customers. I have seen the judgment as questionable. Okay, it's not right, but it's even safer, let's say. So it's questionable. I have seen these three judgments: satisfactory, unsatisfactory, and questionable. Another question is, how do you assess precision for one assay? Yeah, we did not describe we did not enter, but precision is a portion of the validation. We focused more on the CC beta aspect that we have learned that is not very easy. While precision concepts are in common with many other techniques. So basically, Francesca, correct me if I'm wrong, we do perform the so-called intra-assay precision assessment, where we typically run replicates of the same extract within one assay. And this gives the value of the precision of the assay itself, right? And then we try to insert the higher variability we are able inside our laboratory. So the intermediate reproducibility, so-called inter-assay precision, is the repetition of the extraction, first and foremost, typically involving more than one operator through different days. So the environment has changed the maximum way possible, I mean, considering that we are in one laboratory and we have repeated the assay. So it gives a different degree of information over precision. If it is possible, yeah, also the interlaboratory trials provide the reproducibility of the assay. And this is something that we try to participate when it is possible. But basically, inside one laboratory, we do these two measurements: intra-assay, repetition, and more wells of one extract, and intermediate reproducibility. We repeat the extraction day 1, day 2, day 3, trying to involve at least 2 operators and running separate independent sessions. I think, Francesca, we covered. Okay, I think you're muted. Okay.
Yes.
Thank you. Okay, very good. I think we are out of time. Thank you for those that remained connected. For further questions, do not hesitate to contact us at this email address. Thank you again and have a nice rest of the day.